Welcome to The Explainer. Today, we're taking a fascinating deep dive into molecular biology to explore what I like to call the cellular symphony. We're going to unpack the incredibly elegant mechanisms behind gene expression regulation. It's an absolutely beautiful system, and I am so pumped to guide you through this masterclass. Let's get right into it. Okay, let's dive into this. Think about it. Every single cell in your body contains the exact same manual, the exact same DNA, right? So why on earth does a neuron look and behave completely differently from a muscle cell? Or how does a simple bacterium instantly adapt to a sudden change in its environment? Well, the secret doesn't lie in the genes they have, but in which genes they actually use. To really get this, we need to nail down a definition for gene expression. It's basically the complete set of processes that allow a gene to be transcribed into RNA and then translated into a functional protein. Now, not all genes are playing the same game. We've got housekeeping genes. These are your baseline survival genes, just always on, chugging away in all cells. But then we have regulated genes. These are dynamic. They're only active at specific times in specific cells, reacting perfectly to exactly what the organism needs in that exact moment. It's all about timing. Here is our roadmap for today's explainer. First, an introduction to gene expression. Second, operons and prokaryotes. Third, the lactose operon model. Fourth, the tryptophan operon in detail. Fifth, regulation in eukaryotes. And finally, number 6, the multiple levels of control. So, Section 1, Introduction to Gene Expression, the basics. Moving right along to Section 2, operons in prokaryotes, the master switch. We're going to look at bacterial systems first. These guys are incredibly elegant, highly efficient survival machines. And this efficiency comes from something called an operon. An operon is a functional unit of clustered genes, all under the control of a single promoter. They are co-transcribed into one long strand called a polycistronic mRNA. To put that in plain English, bacteria just group related genes together. Instead of turning on 5 different genes individually, they flip a single master switch to activate an entire metabolic pathway at once. Talk about a time saver. Notice the smooth layout here. Starting at the very beginning, all the way on the left, we have the promoter. That's where the transcription machinery actually binds. Embedded right next to it, or sometimes even within it, is the operator. That's the actual switch. And then, reading from left to right, we have all the structural genes, marked E through A. Because they sit right next to each other on the chromosome, the cell literally reads them as one single contiguous unit. It's biological efficiency at its absolute finest. Section 3, the lactose operon model. A Nobel-winning discovery. Now, what's really interesting about this elegant system is that it's our primary case study for an inducible catabolic pathway. And actually, figuring this out won scientists Jacob and Monod the Nobel Prize in Medicine back in 1965. What's so fascinating is how physical this entire process is. If you focus right on those red structures in the center, you'll see the Lac-I regulatory gene produces a repressor protein. It's that 4-part tetramer, and it literally locks onto the operator site marked O. When this repressor sits there, it acts like a physical roadblock. The green blob there, the RNA polymerase, it simply cannot move forward to transcribe the structural genes LAC-Z, Y, and A. So the pathway to digest lactose is firmly shut off to save energy. But wait, there is a whole second layer of control here. Let's focus on the first scenario, transcription repressed. When glucose is highly available, the cell absolutely prefers it because, well, it's easy energy. The presence of glucose keeps the levels of a signaling molecule called cyclic AMP, or CAMP, very, very low. Look closely at the P promoter region. Because CAMP is low, the cap protein, which usually boosts transcription, cannot bind to the DNA. It just floats around unbound. So even if our roadblock was gone, the engine wouldn't have the key to start effectively. Let's move to the next scenario. What if both glucose and lactose are present? Well, the lactose, specifically an isomer called allolactose, binds to that red repressor roadblock, changes its physical shape, and plucks it right off the DNA. The path is clear. But because glucose is still high, our cap protein is still unbound. The result? The green RNA polymerase binds, but only very weakly. We get what's called basal transcription. It's just a tiny, minimal trickle of enzyme production. The cell is essentially saying, sure, lactose is here, but I've got glucose, so I'm not going to waste energy fully turning this factory on. This brings us to a beautiful logic matrix. Let's walk through this slowly. For the Lac Operon to be fully active, which is that bottom row of our table, 2 strict Non-negotiable conditions must be met. First, glucose must be absent. This causes CAMP levels to spike, allowing the CAP protein to bind and act as an accelerator pedal. Second, lactose must be present to bind to the repressor and pull away that physical roadblock. Only when the accelerator is pressed and the brake is removed does the operon fire at full capacity to digest lactose.
Section 4. The tryptophan operon in detail. Anabolic regulation.
Okay, we're shifting gears now. The Lac Operon was all about breaking down food, right? But the tryptophan, or Trp Operon, is about building something the cell actively needs. The goal here is to manufacture the amino acid tryptophan using 5 structural genes. But look at the bottom half of the diagram, where tryptophan is actually present. When the cell has produced enough tryptophan, the tryptophan molecules themselves bind to an inactive aporepressor. This binding activates the repressor, allowing it to lock onto the operator and completely shut down the factory. It's a perfect biological feedback loop. Tryptophan essentially acts as a co-repressor of its very own synthesis. This guarantees the cell never ever wastes energy making more than it needs.
So the crucial point is this fundamental contrast. Prokaryotes, like bacteria, rely on immediate, rapid environmental adaptation. They use operons as simple, highly efficient master switches to survive from moment to moment. Eukaryotes, on the other hand, require complex tissue specificity. We rely on highly independent, layered gene control to manage incredibly complex multicellular life.
Section 5. Regulation in eukaryotes. The cellular symphony.
If bacteria are simple on-off light switches, eukaryotic cells are like massive, complex studio soundboards with dozens of sliders and dials. The very first barrier in our eukaryotic soundboard is physical access. Our DNA is tightly coiled into chromatin, so it's got to be uncoiled first. But even then, there's a chemical lock. Following the chemical reaction diagrammed here, enzymes called DNMTs add a methyl group to cytosine bases on the DNA. This methylation acts to basically silence the gene. So for transcription to occur, the gene must not only be physically accessible, but generally unmethylated.
Section 6. The multiple levels of control, a layered defense.
Once we get past that initial physical and chemical lock, the eukaryotic cell puts the genetic blueprint through a rigorous multi-step gauntlet. Let's outline this gauntlet. First, gene activation, which we just discussed with chromatin and methylation. Second, the heavy lifter, transcription regulation. Third, mRNA maturation. Fourth, translation control. And finally, post-translational control. Let's see how these next layers actually work in practice. This brilliantly illustrates level 2, transcription regulation. Just look at this massive assembly of proteins. The gray structure on the right is the RNA polymerase II, sitting at the start line. But it can't just run, it requires cis-regulatory sequences built right into the DNA, like the famous Tata box sitting next to it. Further upstream, we have enhancer sequences in red and blue. These interact with transacting factors, transcription proteins that bind to the DNA to act as molecular volume dials. They either speed up or severely inhibit the recruitment of the RNA polymerase. It is a highly coordinated chemical dance. Once transcribed, we reach level 3, mRNA maturation. Look at the distinct blocks of color here representing exons. Those are the coding regions. Eukaryotes can perform what's called alternative splicing. By choosing to include or exclude certain colored blocks, like the red exon 4 you see splitting off into 2 different pathways, one single pre-mRNA strand can be spliced together in multiple combinations. This means one single gene can produce entirely different mature mRNAs, and those eventually become entirely different proteins. Mind-blowing, right? At level 4, translational control, things get even more specific. Take a look at this incredible example with the apolipoprotein B gene. The exact same mRNA transcript is produced, but in the intestine, a specific RNA editing process alters a single letter, changing a C to a U in that yellow box. This creates an early stop codon. So, while the liver translates a massive 4,500 amino acid protein to transport cholesterol, the intestine translates a much shorter 2,100 amino acid protein specifically designed to absorb lipids. The exact same gene yields a totally different functional output based entirely on tissue location. Finally, we have post-translational control. This is modifying the folded protein itself to turn it on or off as needed. As you look at this stunning 3D molecular structure of proteins physically interacting with the DNA double helix, just think about the immense scale of what we've covered today.
We went from simple bacterial operon switches, maximizing survival in a puddle of water, to the layered, multi-step gauntlet of eukaryotic regulation, ensuring that your heart cells beat and your liver cells filter. It's this intricate molecular dance that sustains the sheer complexity of multicellular life.
Which leaves us with a truly fascinating thought to ponder. If a shifting environment can instantly toggle bacterial switches on and off, and if things like our environment can chemically alter our own complex eukaryotic DNA through methylation, how might our daily choices right now be rewriting our own cellular symphony? Thank you so much for joining me in this explainer today. Keep exploring, keep asking questions, and above all, stay intellectually curious.